Let me introduce myself first because I'm new to this forum. I'm Sanjeeva Nayaka. I work as Chief Scientist here in National Botanical Research Institute. I'm a taxonomist. I work on lichens. So in this session we have three speakers. The first plenary speaker is none other than Professor H.A. Ranganath. So it is my privilege to introduce him to you all. Most of you all know him, but the sake of juniors, the students, let me read few lines at least about him. So he is a distinguished geneticist. He did his PhD from Mysore University. He served as Vice Chancellor in Bangalore University and Director of NAC. He is a fellow of IIS. Okay.
First of all, I have to thank the organizer for inviting me to give a talk in this session. So my topic I have chosen is neo-sex chromosomes in Drosophila. And as you could see now, this is my team, what we have done at my University of Mysore. And subsequently, there is one more group is there, Doris Bracknog at the University of Berkeley, California, and they are not the co-authors. But what I have done is I have drawn much of the information in the last 10 years, what they have done, particularly on the model which we had developed. Therefore, not to show that they are co-authors, but to share that much of the information I have drawn from them, and I'll just to give an overview of what we did at Mysore and what they are doing at University of California, Berkeley. And as the title you could see, neo-sex chromosomes in Drosophila. So basic information for students, and as you could see that there is always karyotypic dimorphism in animals. Karyotypic dimorphism, that only to say that sex chromosomes are different between males and females. Phylogenetically, we derive all these sex differences chromosomes from a pair of autosomes. But during the course of evolution, many things have happened, and so-called homologous chromosomes, autosomes, over a period of time, on an evolutionary time scale, they differentiate. And some of the events that promote this differentiation are like this. And to start with, one of the genes will start identify itself to recognize the male, and that particular chromosome over a period of time stops recombination with its homologue. When it stops recombination with the homologue, many other changes would happen. As you could have seen, the Y chromosome in many species of Drosophila, or even including human beings, that Y chromosome is degenerate because many things happen because of mutations, and they cannot escape because there is no recombination. Now the accumulation of transposon, so many things would happen to this chromosome which defies meiotic recombination, finally degenerates. And one consequence of this is now, and now people are trying to understand how these sex differences have happened between X and Y. And most of the model systems we have is an X chromosome and a Y chromosome is completely degenerate, and once it has degenerated, compensated dosage compensated mechanisms have appeared on the X chromosome. But today we have the final products of evolution. But over the last 20 years, people have tried to understand how this process was initiated. Is it possible to trace them over a period of time? And Drosophila has given a good hand to do this, and we have a system called Neo-X and Neo-Y chromosome. During next 30 minutes or so, I will try to go through this. One peculiarity of Drosophila is that Drosophila males don't have meiotic recombination. Of course, one of the model systems, what I am likely to speak later, has meiotic recombination. But in general, we would say that Drosophila males do not have meiotic recombination. So any chromosome which is restricted to the male genotype, it cannot take part in the meiosis recombination. The one such chromosome is the Y chromosome or the Neo-Y chromosome. And many people over a period of time, Luchesi and others, Bracknog and others, have shown how these things have happened and what are the consequences of this degeneration. And now why we have chosen this model is because of the relatively recent origin. Neo-X and Neo-Y chromosomes provide useful models to understand the different trends in the evolution of sex differences between X and Y. So there are some already well set examples. I have taken 3 examples. Of course, the first one is Drosophila basic karyotype, which has 2n equals 12. And we can trace the karyotypes of all the existing Drosophila species from this basic karyotype. And how is this possible? Because all these Drosophila species have polytene chromosomes. Through analysis of the polytene chromosomes, we can establish the homology or the differences wherever fusion, fission, or translocations and inversions have taken place. Because of such advantage, even today Drosophila virilis is there, which has 2n equals to 12. And with the help of polytene chromosomes, we can show the relationship. And now one such important step here is the evolution of pseudo-obscura. Rabzhansky and others, many people have worked on that. And in this pseudo-obscura, as you could see here, this is one well-established system where chromosome degeneration has taken, that is, Y chromosome degeneration has taken place, and correspondingly, the entire X chromosome has dosage compensated. That means it has become hyperactive. The hyperactivity is one strategy in dosage compensation, whereas in human, dosage compensation is there by another mechanism, whereas in females, one of the X chromosomes will be lionized or inactivated. There are 2 different strategies. Here, the age of Drosophila pseudo-obscura is about 60 million years. During this period, the entire Y chromosome, which is restricted to male, has suffered degeneration, and only few genes remain active which are involved in sex differentiation. So consequently, to compensate the difference between the male and the female, particularly X-linked genes, the male has only one set of X-linked genes, and now to compensate that, the corresponding X chromosome has dosage compensated. So now we could see here in the next species, Miranda, which is derived from pseudo-obscura, the age is just 1 million years. And now degeneration is in the process, but it is not completely degenerated. Correspondingly, what has happened is Y chromosome is incompletely degenerated, and still there are many genes which remain active. Consequently, what has happened is dosage compensation has taken place on the X chromosome in the male of Drosophila, but this dosage compensation is restricted to those regions where in Y chromosome degeneration has taken place. Therefore, incomplete degeneration and incomplete dosage compensation. This is transient phase. And next stage, you could see here, there is another species called Busquet. Busquet is still younger. Here, it is about 0.8 million years. 60% of it is much younger than Miranda, and here the degeneration of the Y chromosome is there, but not, it is incomplete again, and it is not to the extent of what we have seen in Miranda. So like that, you could see total degeneration and total compensation in pseudo-obscura, even in Melanogaster, and then Miranda, incomplete degeneration and incomplete dosage compensation, and finally in Busquet. These are the 3 stages what we have so far. And people wanted to learn still earlier stages how these things are initiated during the course of evolution. So for the evolution of the dosage compensation mechanism, one of the prerequisites is degeneration. That is, any chromosome which is restricted to the male genotype, because there is no meiotic recombination. Consequently, so many things has happened and it will degenerate. So within the genus Drosophila, there are naturally occurring variations in the sex karyotype. The length, this is more important, the length of time since the fusion event has created the Neo-X and Neo-Y chromosome arms correlates with the extent of degeneration. That means there is a strict correlation between the time of degeneration, the extent of degenerations, and the time elapsed after this origin. And they wanted an N chromosome, so it is of great interest and to examine a very young Neo-Y chromosome where there has been not a initiation of the degeneration, and therefore one can study over a period of time if at all there is recombination, can we start from the beginning and to understand how recombination is taking place? And that's why we have one such species, but it's called albumicans, and taxonomically, phylogenetically, its age is less than 100,000 years, much younger than other species what we have. The young age of albumicans, its large population size helps us to study many of the things that are happening in this particular species. And that's why we call it as nascent neo-X chromosomes in Drosophila, that is albumicans. And this is a potent model system to study the very, very early stages of sex chromosome evolution. Of course, some more advantages are there, but I will restrict myself to the sex chromosomes. So this is in brief how the evolution of the karyotype has been traced. As I said earlier, the basic karyotype is 2n equals to 12 with the first one, and gradually centric fusions and pericentric inversions have taken place to yield one species called Drosophila nasuta, another one is called this albumicans. Drosophila nasuta has 8 chromosomes and Drosophila albumicans has the 6 chromosomes. This is the lowest number of chromosomes that one can see in Drosophila, and 2n12 is the highest. And we can establish the homology of all these chromosomes with the help of polytene chromosomes. And now, what we are interested is in this particular species, that is 2n equals 6. During the evolution of this 2n equals 6, what has happened was there is a fusion. There is a centric fusion between an autosome called chromosome 3 and the sex chromosomes. So the chromosome 3 has undergone fusion with the X, and also the chromosome 3 has undergone fusion with the Y. So there is neo-X and neo-Y, and because of that, the chromosome number is reduced to 6. In the next cartoon, I will show you this now. So that is the basic karyotype. And this is the 2n equals 8 of nasuta, and that is the 2n 6 of albumicans. In the next slide, you will see how these things have happened. So this is the basic karyotype Drosophila nasuta with 2n equals 8. Now you can see now, this is albumicans. The second chromosome and the dot chromosomes are not involved. The second and the chromosome 4 is not involved, but the chromosome 3 and the sex chromosomes have undergone fusion. This is the one, and now, okay. So this the X chromosomes and the chromosome 3 has undergone fusion, and as you could see here in the male, you have X3 and Y3. The chromosome 3 has fused with the X chromosome that has transformed into neo-X, and simultaneously the chromosome 3 has fused with the Y chromosome, therefore it has become Y3. And again, even though the chromosome number is different, 2n equals 8 and 2n equals 6, technically there's no difference in the polytene chromosomes. If you analyze the polytene chromosomes, you will not be able to detect whether it is from nasuta or whether it is from albumicans. They are totally homosequential. So now, this is actual chromosomes. You could see here, this is the neo-X and that is the neo-Y, and these were independently existing in the parent species, but they have undergone fusion. This is what is called a centric fusion leading to the formation of neo-sex chromosomes. And the upper one is the nasuta, the lower one is albumicans. And you could see here in female, there are 2 neo-X chromosomes, and whereas here one neo-X and one neo-Y. The last line, the product of centric fusion between an autosome and the sex chromosome has resulted in the neo-sex chromosome. It is not completely sex chromosome now still, because 50% of the karyotype is still euchromatic. Now the next one you could see here, Drosophila nasuta has a complete pair of heteromorphic XY. See the XY in the figure you could see they are totally heteromorphic. One is X, another is completely heterochromatinized Y chromosome. And in albumicans, now please note the difference now. In this albumicans, it has 2n equals 6, a pair of metacentric incomplete heteromorphic chromosomes. Incomplete heteromorphic in this male because in the neo X, you have X chromosome is fused with the chromosome 3, and this is the chromosome 3. And whereas the chromosome 3 has fused with a Y, that has become neo Y. When you compare these two things, and also you can compare it with that of the Nasuta, it is not completely heteromorphic because this particular arm, the third chromosome 3, is almost homologous to the chromosome 3 of the Y chromosome. That means 50% of these two chromosomes still they have retained homology. Therefore, this is a case where the complete heteromorphic nature is not there in the sex chromosomes. Of course, this is the karyotype of the females. Again, I have repeated, the neo X chromosome has two euchromatic arms. You could see here, this is one euchromatic arm, another is euchromatic arm, representing the ancestral X, while the other chromosome represents the chromosome 3. In the metacentric Y chromosome, that's what I said, you could see here, has one arm with a complete heteromorphic ancestral Y chromosome. The darkly stained part here is the original Y chromosome, whereas the other arm, I have labeled it as YR3, is euchromatic. So when compared to the Nasuta, you could see that the X chromosome has two euchromatic arms, whereas the neo Y chromosome has one euchromatic and one heteromorphic. The heterozygote degenerated, or it is heterozygote inversions. And now, as I said already, in contrast to other Drosophila males, but Drosophila Nasuta males recombine even today, whereas the species which is derived from Nasuta, that is albumicans, today there is no recombination, achiasmy. That's what we had decided earlier. But next in the 15 minutes I will tell you, even during the evolution of albumicans through centric fusion, in the initial phases there was recombination, and how did we prove it? We'll come to that. So these are the important features. So albumicans is the young species that recently diverged from West African species of Nasuta, but the age is about 150 to 500,000 years. That is fairly young now. Of course, these are some of the comparisons to show. This is the albumican of male and the female. The unique feature of this albumicans is nearly 60% of the genes are sex-linked, because two chromosomes have undergone fusion, chromosome 3 and Y, chromosome 3 and X. So in the entire genome, 60% of the genes are sex-linked now. And in addition to that, 50% of the Y chromosome, 50% of the neo Y chromosome is euchromatic. These are some of the exceptions which has led to the evolution and which also has an impact on the dosage compensation. And as I said already, it is homosequential. If you hybridize, all the chromosomes pair synapse without any difference. We call such species as homosequential. When we have hybridized Nasuta and albumicans, we have got this F1 slide only to convince you that they are homosequential. Metaphase karyotype is different, but at the polyethylene chromosome level, they are homosequential. Now, we have studied the dosage compensation in Nasuta and albumicans to start with autoradiographic studies. And of course, from India, Mukherjee, Bierman, Lakhotia and his group, all these people have worked on melanogaster and also on pseudoobscuron, bipectinata, and all these people have shown the total dosage is decompensation because the Y chromosome is degenerate and the X chromosome is exhibiting dosage compensation in males. And how it they have measured, next slide I will tell you, in males, faster replication of the X chromosome. Why it is faster replication? Because its counterpart is not present. The one which is used to the Y chromosome, it is totally degenerate and it is heterochromatinized and it is in the chromocenter. So technically, quantum-wise, the X chromosome in the males is only one copy, and therefore it undergoes faster replication. And another important feature of dosage compensation is hypertranscription. Why this is happening? In males, the sex-linked genes are present in one copy, whereas in females, sex-linked genes are present in 2 copies. To compensate this, what has happened is, in this insect, one of the evolutionary strategies is to make one X chromosome in the male hyperactive, matching the product of what 2 X chromosomes are doing in the females. These are 2 important features that one can look into it. Of course, these are autoradiograms to only show the replication studies we have done. In brief, absolutely, there is no difference between, if you study the autoradiograms of Nasuta and albamicans, replication-wise, the original X chromosome here and the X chromosome here, they are faster replicating. But the other arm which has undergone fusion, the chromosome 3, which is associated with the Y chromosome, still mimics its properties of an autosome. As I said earlier, chromosome 3 was an autosome and it has fused with the X and Y. This autosome 3, even after fusing with the Y chromosome, still it is mimicking the properties of an autosome. So even though this particular chromosome is restricted to the male genotype of albamicans, it has not undergone any change. So only this slide to show you that the fused products still have retained their original autosomal characters. Similarly, when you study the transcription, again, the message is the original X chromosome has undergoing hyper transcription in the male, whereas the other chromosome, which is fused with the Y chromosome, it has not undergoing any hypertranscription or more quantity of RNA synthesis. So even today, the chromosome 3, both in males and females, reflect the autosomal characters, even though they are a part of the sex chromosomes. That means what I have required is not dosage compensated, and this system is yet to acquire the dosage compensatory mechanisms. There are some molecular mechanisms to make sure that single X chromosomes will be hyperacted in other species, but in this species, even though they are a part of the sex chromosomes, dosage compensation is yet to occur. And these are the 3 publications coming from the BRACTA group from Berkeley, and most of the things what I speak now is drawn from the findings of these people. And what they have done is, with the molecular biology, with the genome-wide polymorphism data, complex evolutionary history of new sex chromosomes have been traced. That means the entire genome of Drosophila albamicans, Drosophila nasuta is sequenced, and they have compared it. And some of the decisions have been made, and they are like this. I use this figure again and again only to tell you the karyotype of albamicans, as I said, derived from nasuta. Again I repeat, this ancestral feature of the recombination in males was also seen in the first few years in albamicans. Because albamican males today don't have a recombination, but in the early days of its evolution, these people have shown that there was recombination in the albamicana karyotype of males. How did they do it? Because the molecular data revealed that, of course, they have selected 27 loci on the X and the Y, and there is a lot of sharing polymorphism between the X and Y. This shared polymorphism was possible only if there was an exchange of material between the X and the Y. So later on, at a later date, another mutation has taken place to stop the recombination. But in the early days, this was the analysis, the high levels of shared polymorphism between neo-X and neo-Y. See, third chromosome has, I have to repeat, third chromosome has from nasuta, and this nasuta is with the X and the Y, and these 2 chromosomes have shared polymorphism. And what the model says, in the early days of albamicans, there was a recombination, and with that recombination, this polymorphism could be explained. This is what I am trying to show you. So during the initial stages of the evolution of albumicans, males had meiotic recombination. It meant recombination occurred between neo-X and neo-Y in the males of albumicans like that of its parent, that is Nasuta. So now, the chromosome 3 has fused with the X and Y. Right? Now what we have to realize is the chromosome 3 is fused with ancestral Y, it becomes neo-Y. So what we have to remember now is the neo-Y chromosome has a haplotype of the chromosome 3 of Nasuta. Haplotype means only one copy, haploid. So the chromosome 3 is fused with the ancestral Y and it becomes 3Y, neo-Y, and it is a haplotype of which represents its Nasuta ancestry. See now, Nasuta residual haplotype on the neo-Y chromosome is broken down. Now, as I said, already there is a recombination between neo-X and neo-Y. This haplotype undergone recombination. Whatever sequences were present in the haplotype is changed now because of recombination. So it means from an evolutionary point of view, the real haplotype of 3 is exclusively present in Nasuta. Even though it has come down to albumicans, because of recombination, it is no longer mimic the same sequence of genes, same sequence of material of Nasuta now. The recombination with the neo-X generated multiple neo-Y haplotypes in geographically restricted. So this is more important. So these people have collected Drosophila albumicans even from western, northeast part of India, across Japan and Japanese islands, and they could see that this haplotype, particularly the third chromosome part of the neo-Y haplotype, differs in these 3 different regions. That means haplotype has undergone different types of recombination in these geographically restricted areas, depending on that 3 distinct groups. For example, this. The Taiwan and Japan populations have one type of recombination. Okinawan Island and the third one is Shillong and Kunming, that is northeastern part. So within albumicana population, recombination has taken place in different ways, in different frequencies in these 3 different populations. Therefore, they have marked 3 distinct haplotypes now. These 3 distinct haplotypes have come in handy to them to study further studies of evolution of the Y chromosome. So now, all this time I said there was a recombination in the male of Drosophila and also males of albumicans. Suddenly they have recognized a mutant has taken place in the lineage of albumicans, and with that, because molecularly they have shown the mutation, with the arrival of that mutation, the recombination is stopped in the males of albumicans. So this is a basic breakthrough now. Nasuta is an exception, having recombination in males against the absence of recombination in other Drosophila species. Even now albumicans, it started its journey with recombination, but due to a mutation in the recombination genes, even here the recombination is stopped. Again, the molecular data shows the nature of that mutation which inhibited the process of recombination. With that, now, as I said, one of the important consequences of cessation of recombination is gradually mutations happen, and these mutations cannot escape through recombination. They accumulate, and of late people have shown whatever transposons enter into the Y chromosome, they cannot escape. They will also pseudogenize whatever genes that were active. Like this, many things happen over a period of time, and finally it acquires the characters of a heterochromatic. In particular, authors have identified 3 neo-Y haplotypes of different age that are geographically restricted, which I have already told you, like this. So, the advantage is they can look into the Y degeneration, acquisition of recombination, and the acquisition of dosage compensation in these 3 different populations, which are one away from another. There is no crossbreeding, and now they are looking at the degeneration of Y and also the dosage compensation in these different populations. So this raises the possibility that mutation causing the cessation of male recombination albumicon has occurred, and now sequential degeneration is taking place, and that is the information now. However, once male recombination stopped in a particular population, the diversity of the neo-Y gradually, rapidly decreases, and now a new Y chromosome is going to, yeah. So this is how they were recognized. So now people have estimated through molecular biology different times point at which recombination was stopped. Accordingly, the degeneration is taking place. Of course, now you could see here what are the things that are promoting degeneration is many of the genes present on the Y chromosome now they are down-regulated. And a few genes have undergone mutation, or what they have called it as, they have become pseudogenes now. Still, there are many genes which are active, but the initial stages are pseudogenization of a few genes as well as the arrival of many transposon material, and gradually they are moving to. These are the very, very initial stages which is reported for the first time to show how Y chromosome is being heterochromatinized. Suggesting that means it is age related. There are 3 populations of Y chromosome. The extent of degeneration also depends upon the age of that particular species. That means here again, when once mutations are happening there in the Y chromosome, at least a few genes on the X chromosomes are also showing dosage compensation. So there is initiation of degeneration, initiation of acquisition of dosage compensation on the Y chromosome, on the X chromosome. So with all this, one important thing that emerges from this study is testis biased genes escape degeneration. For example, you can look into human Y chromosome, Drosophila Y chromosome, it may be degenerated, it may be heterochromatinized. Yet, during evolution, there is one common trend. That common trend is any gene which is required for male functioning is not touched. Even that's why I put exclamatory mark in the question. Even in albumicans, even in Asuta, even in melanogaster or human, on the Y chromosome today, even though we say it is degenerate, a particular part of the Y chromosome is spared, and that particular part carries some basic information required for maleness. So here also, in the midst of all this degeneration process that is initiated, it has not been touched. So across different karyotypes, this trend is, even today, it is retained. So this is another one, last one. This is another neo-Y chromosome, what we have under the laboratory conditions. You could see now. This experimental evolution is still in progress. It has passed about 20 years of breeding and so many thousands of generations. In one of the population, you could see now, please focus on these arrows. This is the first one is the Nasuta karyotype, and we have hybridized it with albumicans. And after a few years, this new race was formed. That is, we called it as cytorace 1. And please follow the blue chromosome. That is, in the parent, it was chromosome 3, it was an autosome. But by some meiotic mechanism, these stabilized races were evolved. Here, in this stabilized cytorace, the blue chromosome, which was originally 3, which was an autosome, now it is restricted to male genome only. It will never appear in the female genome. For example, this male produces 2 types of gametes, and the female produces 1 type of gamete, and the fusion gives you this male and the female. So only thing to emphasize is the one which was autosome in the parent, in the particular cytorace, it is restricted only to the male, and it never gets into the female. So like any other Y chromosome, this recently evolved neo chromosome also likely to suffer in the due course of evolution, the absence of recombination, etc., and it is likely to generate. So this is how, under laboratory condition, this neo Y chromosome is, what all I spoke so far was naturally evolved, and this is what we have evolved under natural laboratory conditions. And age of this neo Y chromosome is less than 3 decades. because we know when this race has evolved and how this Y chromosome is. Somebody has to start looking at the fate of the genes present on this blue chromosome. So blue chromosome can be compared with the parent Nasuta chromosome, and in the cytosine one and what is happening. It's much more younger than what we had earlier. So Orbimicron has become a powerful system to study and uncover the very, very initial stages of evolution of sex chromosomes, particularly degeneration of Y chromosome and dosage compensation. Thank you.
Thank you, Professor, for a nice and informative talk. And because of the shortage of time, we can ask one or two short questions quickly. So any questions here, please?
Hello, sir. Good afternoon. It was excellent talk and really amazing works. Wondering the geographic location between the Shillong and Kunming, it is I think in the same range. Do that geographical or environmental factor play any important role?
Tell me again.
In
Three
your
populations are there, yeah.
sorry?
Three populations are there, Orbimicrons.
In one cases you are the Shillong and Kunming of China.
Yeah,
They are in the same geographical region.
they are in the same stream,
Yeah.
but there is no interbreeding among those individuals. And also in these three populations, at the molecular level, they have shown when exactly the recombination was stopped. And at different points of time, the male recombination was stopped in these three populations. Accordingly, at different points of time, degeneration has started. So they could compare and contrast these things in these three populations, and also they can compare it with the parent Nasuta, what are the things that are happening. That's why people say that they are one of the best models to uncover the very, very early stages of effect of male restricted chromosome. Thank you.
Thank you, sir.
Thank you very much, sir.